I've been asked to introduce myself for those of you who don't know me, which is, I think, almost everybody. Thank you for coming. I heard somebody enthusiastically say, well, I'm here because it's ticks. Yay, today is ticks. And welcome to the people in Riverside. I am the one of the co-leaders on the Center of Excellence project and the grant in UC Davis, and my labs area obviously is the tick part of it. And although my training and my expertise prior to getting involved in this rickettsia epidemic was primarily Lyme disease and mostly anaplasmosis, which I still regard as my favorite disease, I was sucked into the world of Rocky Mountain spotted fever. I'm really glad I have been. And so today I want to talk about, actually we have 3 interrelated projects. I'm only going to talk about 2, and I can mention the third where that's taken out. And it's work that was just submitted last week for publication. We submitted it to the American Journal of Tropical Medicine and Hygiene. And so I'm pretty excited to talk to you about it. And I'm a very informal person, so if there's questions as we go, you're welcome to interrupt, or we can, I should finish up in plenty of time when we should have a discussion at that point. Oh, does this need an explanation? Yes. These are brown dog ticks covering the inner ear of a dog. And they actually are all adult ticks. They're just adult ticks in different stages of having been fed. So these guys are basically fully replete, and they're going to fall off pretty soon. These guys are working on being replete. I don't quite understand why they geographically segregated themselves the way they did. It's sort of an interesting problem. As Bob and I were just talking about, all of this just keeps raising really interesting questions. So it's great. It keeps us in business. Next slide.
Yeah. Is that someone's dog?
Yes.
Did you notice this?
In the face of infestations with ticks of this level, brown dog ticks, some of the amblyomas in the Midwest, there's almost nothing you can do. I've heard of people taking dogs with amblyoma americanum infestations to veterinarians and have veterinarians have to anesthetize dogs to get all the ticks off of them. There's a limit to what you can do. Jamie's going to try to get the next slide thingy going for us. Not my computer. I don't know. It might be. Is it because of the sharing stuff or?
Well, I have
yeah, sure.
Do ticks feed at different rates depending on the blood? Next slide.
No, go ahead.
The geographic distribution of the ticks on the ears from someone who knows nothing about ticks. Like if the ones are closer to higher blood flow, maybe they're able to feed faster than the ones.
Bob, you can weigh in if you know an answer to that. I have never seen anybody really talk about that. Usually, for a given species and a given ambient temperature and humidity and host, they tend to be, depending on the tick stage, they tend to be on for a fairly set amount of time.
Okay.
So like you can pretty much predict that an Ixodes pacificus larva is going to feed for, I don't know, 4 or 5 days, and then a nymph 5 or 6. You know what I mean? Okay. Jamie, click on this picture and see if we get what I want to see happen there. And if it's too much, then Jamie's like, wait a minute, why did I get this job? There you go. These are brown dog ticks, so if you know, there's people, for the Riverside people who don't get to see this, there's people in this room scratching themselves. Awesome. Those are brown dog ticks. I just want to point out that the temperature at the time that this was happening was 120 degrees. This is in Mexicali. So this is hot. This is, for those of us who've worked on these coastal, cool weather, high humidity desiring ticks, this is really, really hot. And yet, when we get to a home, we look at the bare ground up the adobe walls. That's what this tick is. It's an arid adapted tick that's found, when I say worldwide, it's certainly not in Antarctica. And it's, patchily, even in California, where I've seen it in the valley, especially in animal shelters and in kennels. So another name for it is the kennel tick. And it, as the name suggests, it's a sensulato, as we're discovering, next slide please, as we were discovering, the brown dog tick is a species complex of ticks that are morphologically non-differentiatable. They're all identified by this unique shape to the head, the fact that they do have festoons and an inornate sputum. But when you do genetic work, behavioral work, other kinds of work, you can detect several different species. And within the genus Rhipicephalus, full hybridization across species is a phenomenon. It's important. All stages of Rhipicephalus sanguineus sensuato, to our knowledge, prefer to feed on canine hosts, and that's dogs, but also coyotes and other canids. And they are known to transmit diseases, and because they will feed on dogs, that makes them fully peri-domestic. And that's a little unusual for some of our North American tick species. So most of our North American tick species have sylvatic components to their life cycle, where as immatures, they're feeding on small mammals or ungulates that are wild. And so unless your home or your activities take you into contact with a tick in the woods, there's a little bit less of a risk. This tick feeds in all three stages on a dog, and so it just will live in the home. Stalking behavior, you don't need to go back, but stalking behavior, you know, again, being a North American California tick girl, I learned nidicolous, which is living in the nest, and questing. Stalking is what it sounds like. And that's something that some of the arid adaptive ticks in Africa do as well. Okay. And I mentioned that the genus is characterized by hybridization. This paper is one of the best papers I can find that shows a phylogenetic tree that helps me illustrate what I want to illustrate here. Move this cord to kill somebody, I mean myself. They used a soft tick as an outgroup, and then a couple other outgroups. But then within Rhipicephalus, we see Rhipicephalus sanguineus from China. Then we see a group of ticks, this Rhipicephalus tyrannicus, which as a tick that until recently was considered a sanguineus. So already there's a new tick that has a new species. A bunch of tyrannicuses and a bunch of tyrannicuses, and their sister group is sanguineus. Within sanguineus, we know, next slide please, that there's at least two really important strains, and a couple of these pictures are big, so they might take a minute. There's two really important strains, and one of them is in the process of being renamed as a new species. I'm gonna now wait for her. I'm sorry, Jamie. I don't know either.
It's advancing on my screen.
Oh, is it showing on your screen? I'm going to look at it really quickly because I can talk about it as I know. Okay. That one.
Try this one more time.
There we go. Okay. So another tree, this is now looking just at Rhipicephalus sanguineus. And what, I'm sorry, I didn't put the citation for this one on. This is breaking it down into these two lineages, the tropical lineage and the temperate lineage. This is, and then you'll notice there's some tyrannicus in here, some Switzerland and Italy, and probably Nigeria. This is really important. This is a big take-home message for my whole talk. I'll take a minute to make sure we're clear on this one. The tropical lineage has been found in Chicago, Florida, Arizona, Peru, South America, Mexico, etc. Okay? And for the most part, it seems like it's a even hotter adapted tick that is a little more likely to feed on people than the temperate lineage. The temperate lineage is the one that we see in Portugal, Europe, United States. It's been found in Sacramento, SoCal, which is near Santa Cruz, San Diego. It's been found in various parts of the United States. And it's the kennel tick. It's the one we see in hunting dog kennels, animal shelters, and such. Those of us who work on ticks and have for many years went, okay, well that's a three-host tick. It always feeds on dogs. It never ever feeds on people. It transmits Ehrlichia canis, and yeah, I think those are the salient things to its biology. Whereas this tropical lineage, maybe it might be a bias in the literature that we read, but there's a less of a sense that it was, it does feed on people, and I don't think that that's really been explored as thoroughly as it needs to be. So anyway, we've got these two lineages, and genetically they're very differentiatable, but I swear to goodness, they look exactly the same.
Especially for the northern detections of the tropical lineage, what's typical of the detection? Is it typically found engorged on a dog that just moved north?
No, so the temperate one, it's found engorged on a dog in a kennel that has a problem. So they had an ongoing problem for months. Maybe they've even seen some Ehrlichia cases. And maybe they tried to eradicate ticks, but not effectively because they do get up into the cracks in the kennel and the concrete and such.
But for the tropical ones, how do they get to Chicago?
That is probably a dog that moved.
Okay.
I would strongly suspect Chicago is a dog that moved. Okay, next slide, please. All right, so this is a paper that my group submitted last fall, and Zachary Villarreal was an undergraduate student in the global disease biology program. So this was his undergraduate practicum, so I'm pretty proud of that. And what he was asking this question about was looking at this interface between what we know to be tropical lineage Rhipicephalus and what we know to be temperate lineage, and saying, well, you know, is it moving north? So Sacramento was pretty much a positive control for temperate lineage. We actually do have ticks from Oaxaca, which are positive controls for tropical. Just can't show them on the map this easily. In a few minutes, when we talk about Rocky Mountain spotted fever, we'll talk about eastern Arizona. This is temperate. Traditional, old-fashioned United States ticks. We see tropical at the Mexico-California border, but unfortunately, we actually see tropical now in Los Angeles as well, in San Bernardino Mountains in Los Angeles. So for the first time, we actually have evidence of this tick moving north. Next slide, please. So I'm going to switch gears and now talk about the disease, and then we'll put them together. Rocky Mountain spotted fever can present like this. This is a petechial rash. It is caused when the bacteria here, immunohistochemically stained to be red, invade the blood endothelial cells which line the blood vessels. When the endothelial cells are infected with rickettsia, vessels are no longer what we call patent, and so the blood doesn't stay in, and so we start to see leakage of blood out of the blood vessels to cause a clinical syndrome that looks like that in some cases. The bacteria that causes it is Rickettsia rickettsii. There's between 500 and 2,500 cases each year in the United States. And like pretty much all the diseases we all work on, it's a non-specific flu-like illness. Presents within a week or two after a tick bite, and it initially presents with fever and pain. It can progress, not always, to these rashes, causing conjunctivitis and neurosigns. And in a worst case scenario, and that worst case scenario can even be as high as 5%, even with treatment of case fatality. It can progress to respiratory failure, CNS failure, and herniation of the cerebellum, kidney disease, a partial paralysis from which the patient never recovers, gangrene in distal limbs, and they can lose their limbs, and fatality. Okay, next slide, please. And just to quickly put it into context, we are in the family Rickettsiaceae, which is a sister clade to Anaplasmaceae, containing my favorite genus, Anaplasma. Rickettsiaceae and the genus Rickettsia is growing by leaps and bounds as new syndromes are detected worldwide and looked at by molecular techniques, such that currently in the Rickettsias we have these organisms. I don't need to number them all, but you know, there's dozens and dozens of Candidatuses and well-described rickettsial species, and they're transmitted by fleas, lice, ticks, heads, and potentially other arthropods as well. Next slide, please. So sorry about the busyness of this slide. Okay, this weekend, I confess, I went on Wikipedia and I read the article about Rickettsia rickettsii, and it was utterly fascinating. There's a lot of neat historical stuff that I hadn't ever heard. So if you ever want to get into like what happened 100 years ago in the good old names, that was really entertaining for me. So it's originally seen in the late 1800s in Montana, and so that's where it got its name, Rocky Mountain spotted fever. And an astute physician detected a patient, put together that a patient that had this flu-like illness that was starting to emerge in the Bitterroot Valley had been recently fed on by a tick. Howard Ricketts, from which the organism was obviously named, identified the pathogen and also described the basic epidemiology And ironically, he did die of typhus while in Mexico, and typhus is not rickettsia. Researchers, many of the researchers experienced fatality. This is not a safe pathogen to work with. It's a BSL-3, and it's fairly easy to contract and without appropriate biocontainment, which they didn't know anything about at the time. There were fatalities. And clever or brave individual decided to create a vaccine, a crude vaccine, by literally mashing up ticks, inactivating everything with this weak carbolic acid, and injecting it into himself. That is dedication. At the time, it was known or learned to be transmitted by a tick that we have in California. So this tick is typically in the northeast part of California. And as I mentioned, this is a lot of the ticks that we experience here feed, on larger animals as an adult, but as larvae and nymphs, they tend to feed on small mammals. So this would have required a small mammal component to the life cycle. And we know that rickettsias can be transovarially transmitted, but they do impose, depending on which paper you're reading, a fitness cost to the tick that's infected. So you would think that the colony would tend to die out over time. As a fun fact, there have been reports of Rocky Mountain spotted fever in California, including on the West Coast, and we don't actually think that they were Rocky Mountain spotted fever. We think that they were misidentified, that they were viral and misidentified. Okay, next slide, please. So Rocky Mountain spotted fever is emerging in the United States, just not the Rocky Mountains. It has left the Rocky Mountains for reasons I do not understand, and is primarily seen now in the, I don't know what you want to call that, South, Southeast, Midwest, and it's associated with Dermacentor variabilis, and they may know the common name for Dermacentor variabilis. It is the American dog tick. So we've left now the wood tick, and we've moved back into another tick that specializes on dogs. Okay, next. And in 2006, Lee Nicholson at the CDC and other collaborators published this paper, Rocky Mountain Spotted Fever in Arizona, with this tick. And I swear when I saw this hit me in my ProMed, I was like, oh, that's a typo. I literally thought it's a typo. Rhipicephalus sanguineus does not transmit Rocky Mountain Spotted Fever. Well, it does. And so it was a novel tick for Rocky Mountain Spotted Fever in the United States. And these are pictures out of the CDC, so I apologize for the resolution of them, pixelation or something. So this dog has kind of a lot of ticks in his ears. We've got workers with a rabies pole trying to contain this dog so that they can put a collar on it. Because what they were donated collars that have an 8-month effectiveness against ticks. They're acrocidal collars on dogs. So by putting these collars on the dogs and doing aggressive spay-neuter campaigns, because what happens in some of these areas with not great veterinary care for the dogs is there's a high turnover. So the older dogs get hit by a car, or they just don't make it back past a couple of years. There's lots and lots of new puppies recruited. There's no herd immunity. It's not awesome for the dogs. It's in this situation they were overrun with strays, and actually many of the cases were seen of Rocky Mountain Spotted Fever were seen in children who were playing with the dogs. But by the combination of spay neuter and long-acting acrocidal collars, they were able to get on top of the epidemic and actually end it. But as a sort of interesting side note, I've been told it kind of keeps coming back. This is the kind of thing that you have to sustain. You have to keep up the spay neuters. You have to, and they're not being granted the $80 acrocidal collar for free anymore. So it's frustrating because we could get back to the same place we were in 2006 without real sustained public health efforts in these tribal areas in eastern Arizona with the temperate strain of Rhipicephalus. Okay, next slide. Well, it's not true that Rhipicephalus sanguineus doesn't transmit Rocky Mountain Spotted Fever. That is just me not knowing because we have known since 1943 that when Bustamante published in Spanish that there was a new rickettsiosis in Mexico associated with the brown dog tick, and that would have been the tropical lineage. And I like this map. It's published by Alvarez, who's associated with the Sonora epidemic. We're going to show that in a second. He's done really great work in Sonora. This is the state of Sonora up here. This is Baja California Norte. But you know, for color coding, historical outbreaks of Rocky Mountain spotted fever are in dark blue. Rickettsia has now been detected in ticks from Yucatan, Veracruz, middle of the country. The outbreaks have only been detected from about La Laguna, which is in the state that I don't know which state this is, up to Mexicali. Okay. And at the time that Bustamante worked on describing the outbreaks, they were much smaller, much smaller. He actually described them with two clinical syndromes, which is, again, a really interesting question to me. He called them mountain syndrome and the coastal syndrome. And one was much more virulent than the other. So I want to leave us, when we get done with this hour, asking the question of, are the tick strains more or less likely to transmit different strains of rickettsia? Are there different virulence factors in the different rickettsial strains? Is this an environmental factor? Is it a factor of poverty and the people and their inability to seek health care? Why do you see differential health outcomes in these different scenarios? And going back this far in time, I don't know that we're going to have the answer to that, but I think now that we're working on it with modern techniques, we can get to the bottom of that. And next slide, please. So I mentioned Alvarez and his really amazing work in Sonora. If you've had a hard time with the tick on the ticks on the ear, this one's even worse in my opinion. This is a child who is dying, and in Sonora State from 2003, when the epidemic was first detected, through now we've seen more than 1,200 cases with a case fatality rate of greater than 1 in 4. It is typically in an urban setting. And most of the people who die of Rocky Mountain spotted fever in Sonora have what's called Seguro Popular, and I'm going to explain. That's their default health insurance. It's inadequate. It's a Medicare kind of health insurance. It is a short term, and I promise I won't do too much or any into politics, but instead of having good quality health care, some people have this default short term, which is what we're trying to bring back in the United States. And most of us care about healthcare. So when you see outbreaks of new diseases like this, and one of the big things that kills people is delays in treatment because they can't afford to go to the doctor, that's just really frustrating to me, because this could potentially be treatable. And the odds ratio, you were 3 times more likely to die in Sonora if you didn't go to the doctor promptly and have your baby with this. Next slide, please. This was me with a iPhone at a talk that was given in Mexicali. So Mexicali is in Baja California. You'll see another map in a few minutes. And in 2008, so this is 5 years later, we're starting an epidemic in Mexicali. And what's, I think, the most alarming thing about Mexicali is that it's much bigger than Sonora. Much bigger than the outbreaks on tribal lands in eastern Arizona. Like 1.5 million people. When we have 1.5 million people, not all of whom are poor, but many of whom are, oh boy. And the health department and some local academic researchers looked at the city of Mexicali and said, well, where are we seeing cases? 69 cases in this satellite neighborhood, 6 cases here, 25 here, 38 there. So these are peripheral to the city, and they happen to be neighborhoods that are quite poor. Next slide, please. Where is Mexicali? Here it is. So again, this is Sonora State over here. This is Arizona. The outbreaks on tribal lands were in eastern Arizona, associated with the temperate tick, so that's the tick from the rest of the United States. Mexicali is immediately adjacent to the United States. There is a wall that separates Mexicali from Calexico, the state in California, the city in California. What else to say about it? I think that's enough. Okay, next slide, please. So I mentioned that the health department in Mexico has certainly recorded cases. There's some paperwork issues that I don't have time to go into, that certifying a case all the way through the federal government is actually different in Mexico than the way it's done here. Let me talk about that for a second. local county vector controls are often the first place that accumulates the information. That gets transmitted to the state, and the equivalent is tribes. So when tribes have reportable disease, they transmit that information to the state. Then that gets on to CDC if it's a reportable disease. In Mexico, they are not really authorized to do local case confirmation, even though they know it is. Even if they get PCR positives, they still send the cases on to Mexico City for final confirmation. And there's a delay and sometimes lack of capacity in Mexico City to do that final confirmation. But nevertheless, the health department felt fairly confidently that they pretty much knew who was a case and not. And they were detecting cases in especially some of these poor neighborhoods and neighborhoods out in this agricultural valley nearby. And when I happened to be down there getting ready to train our students on how to do our study, there was a little girl who had just gotten to the ICU, and she was from an agricultural home. And we got a chance to go out to her home to see what it was like. And we get out of our car, bare dirt, and we went under a tree, just walked over to a tree. This is 120 degrees, and walked under a tree. There were just ticks crawling everywhere. Ticks carrying a fatal human disease. That was alarming. This little dog was covered with ticks. She wouldn't let us touch her, but she got ticks all over her ears, ticks all the way down her back. And this sat on a dirt floor in the bedroom with the girl that in the ICU. The girl did survive. And so, you know, again, a really interesting question. I didn't do much more with it than that, but to sort of say, you know, what is the exact mechanism by which these ticks are not going to leave this dog. They're attached, they're glued on, they're happy where they are, but their offspring can infest the girl. Okay, next slide, please. So our study design was to, we wanted to get it done quickly because we didn't want this to go over long amounts of time, because then maybe the early sampling dates and later sampling dates might represent different phenomena. So in order to do so, we needed volunteers, and our volunteers came from the UABC, which is the Autonomous University of Baja California, and they happen to have a medical school and a veterinary school. And faculty there were really excited to collaborate, and so they recruited 88 students, all of whom spoke Spanish natively, many of whom spoke English too, and we went door to door. Isa Salud, which is the health department, randomly chose 20 neighborhoods from which there had been a confirmed human case in the last year. And an AHEB is I can't speak Spanish, so feel free if you can. I'm going to butcher this, but it's an area geographica biostatistical basis or something. But we call them AHEBs, and those are their neighborhoods. So we had 20 neighborhoods from which there had been human cases, 20 randomly chosen controls. They were fully randomly chosen. We were blinded as to which neighborhood we were going to. Within them, we picked 5 city blocks, and within each city block, we went to the first home in the Northwest, sought consent. If we didn't get consent, which we often didn't, we went to the next home in a clockwise direction until we got consent. So in the end, we had 200 homes, and at each home, we delivered a very short questionnaire relating to, had you ever heard of Rocky Mountain spotted fever, and did you use Acrocide in your home or on your dogs? We did tick collection, both from the walls, the floors, and the dogs. And ultimately, we did blood collection from the dogs and PCR on the dogs and the ticks. Thank you. We is my undergrads. Alicia, how many ticks did you grind and PCR?
Oh gosh, 400 period.
Emily sort of supervised all of this and did a lot of them too. I don't actually think I did a single PCR this time. Okay, next please. Alrighty. This is a couple of the young women. Typically what happens is the human medical students did the questionnaires, the veterinary students did the dogs. They're coming to a guy, they're knocking on his door, this is Sunday morning, and they're in their white coats, this is all very alarming in this context. And they're like, do you want to participate in a study? Some people said yes. Next slide. Okay. And then tick collection, I'm going to talk for a second about tick collection. Ticks were observed on the walls for a standardized 15 minutes so that we can count them. Okay, so they went in, they started the clock, and then they counted. How many ticks did they see? And then we, collected them. Typically, ticks would start to come out more as they started to smell us. Okay, and then on the dogs, we only collected a set number of ticks, although we ranked the load as none, low, medium, and high. And then we did use the CO2 trap, so we can't use dry ice there. It's just too hot. It was really difficult to get dry ice. It would have cost me thousands of dollars. So this is calcium carbonate and lactic acid, which drips through holes, and then it supposedly works. It didn't work very well for us. So the ticks would presumably be recruited to the smell of the carbon dioxide. It's not sublimating here. It was sublimating from dry ice, and crawl on the rag. We didn't get very many that way, but I'm not sure why. Personally, I think the holes weren't big enough. We did another 15-minute count to see how many dogs there were, and the dogs would be in the home, but also on the street. Because it was trying to get at, remember in eastern Arizona, one of the risks was the overabundance of stray dogs. Okay, and then we did the rest of the work on the dogs. Next slide, please. Okay, so he's examining a dog. The veterinary student. They were asked to wear white and to make sure they tuck their pants in their socks and such, and to check themselves for ticks. Not full compliance. Next slide, please. I love this picture. I don't know why. This is some of our veterinary students. Next picture. Next slide, please. Okay, so our main findings, we're just going to break down dogs, ticks, and people. Overall, 285 dogs tested, and the general prevalence is 67% with some very high titers. Not a single dog was PCR positive. That is common. It's because the pathogen isn't really circulating in the blood. It was initially, but now it's in the endothelial tissue. We do have humans that are PCR positive, but that's because they come in going, I have a high fever, I have a intense pain, I've got a feeling of a rash, just earlier in the infection. Okay? Things that were non-significant for a dog to be seropositive were whether or not you were from a case of Hep, whether you were stray, your body mass, sex, age, acroside. But if you had a poor body condition score, if you were skinny and you were a dog, you were significantly more likely to be seropositive. So I think that's a proxy for poverty and how well the dogs are cared for. Also, smaller dogs were a little more likely to be seropositive. I don't know why. I mean, I guess you can start to imagine when smaller dogs are lower to the ground and those are the ticks. I don't know. And so we put together this in a multivariable mixed model. The final model to best account for serostatus, whether or not you were positive or negative, included your body mass, your age, which I included as a confounder, and then we used a random effect to account for the fact that the ticks, the dogs were not independently cultured. There's multiple amounts. Next slide, please. This is not an atypical picture. I asked, why does the people have dogs? We didn't see a lot of strays. Most of the dogs were owned. Some of them were on the street, but they were still owned dogs. This dog is in a couch, in dirt. That's the size of the yard. It's a guard dog. They have a huge problem with crime, and so a lot of people keep dogs as guard dogs. Keep them outside. They care for them. They feed them. May or may not ever put the money into protecting them from ticks, though. And if they do, it's probably to protect their family. Next slide, please. So the main finding for ticks, we collected almost 3,000 ticks. 12 of them in the end was PCR positive, which is not very many. Okay, but when we broke it down by neighborhood, most neighborhoods had a 0 PCR prevalence, but a few of the very high-risk neighborhoods had prevalence as high as 5%. So 1 out of every 20 tick that would feed on you or your children or your dog could potentially give you Rocky Mouse spotted fever. We did confirm that these were Rickettsia rickettsii by DNA sequencing of the ticks that we could, and epidemiologically, here, the outcome variables, how many ticks we see in the house and dog altogether. If dogs are present, you're more likely to see ticks. That's good. It's kind of, and then all these other risk factors that had been proposed in prior studies didn't come out to be significant for the number of ticks in a home. When we look at the ticks on the dog, I mentioned we had some dogs with no ticks, low, moderate, and high. Being near an agricultural canal is actually a significant risk for your dog to have a high tick load. Again, we don't know why, but the agricultural canals are on the outside of the city and down into the valley. And again, I think it's confounding with the poverty. I think the people in the middle part of the city are just a little bit better off. Next slide, please. So as you often find, if you haven't, for students who haven't done a lot of these kinds of studies, many of your well thought out risk factors turn out to be non-significant. You're lucky if you get a few that are. Okay, when you look at people, what I'm showing here are significant things that are associated with an HEB being classified as a case, a human being having been diagnosed in that HEB in the last year versus a control. And the most important thing that was associated with being a case was having earth substrate. Okay, if you had dirt substrate, that neighborhood was more likely to have case. If you had grass, it was less likely. So there's a protective effect of grass. Okay, it could be the biology of the tick. The tick doesn't love grass, it likes dirt. It could again be poverty. In this neighborhood, you have to spend money to irrigate your home and have grass. Actually, hardly anybody did. And most alarming, almost everyone had heard of rickettsiosis. That's what they call it. They'd heard about it. They knew it was fatal. Even if they owned dogs with ticks, less than half of them used acaricides on their dogs or in their homes. Next slide, please. These were owned dogs, but they're playing in the street. Next slide. Next slide. We did look at some spatial. This is essentially the city of Mexicali. There's a long tail going down the valley. I couldn't get it on the map. These are where our HEBs are, and this is dog seroprevalence. And here you'll see that most of the red, the high prevalence HEBs are on the periphery. Next slide. And the tick loads actually didn't match the prevalence as well. So there's ticks sort of here. I guess I would interpret this to mean that the entire city is at risk. Next slide. Some conclusions and recommendations. Our team, which comprised myself, people from CDC, people from UABC, students, people from Imperial County Health Department, etc., divided up into what we thought was the point of this study. Half of the team, especially the epi people at CDC, said this is an epi study. And a few of us, myself, Chris Paddock, a couple others went, this is a going in descriptive, are there ticks, is there seropositivity, is there rickettsia that we can detect? It was that simple, okay? In either case, these are critically needed data for the local people to make a plan. Obviously, they need more data. They need it on a neighborhood by neighborhood basis. I would argue that doing more dog serology, maybe not so helpful because all of the dogs are positive, but the fact that we now know that we can, unfortunately, you have to test a lot of ticks, but that we can detect really high risk, very focal spatial areas with tick PCR, that's probably what I would do to continue to evaluate risk. Ultimately, this can't go on. We have to get control of this pathogen. It's a treatable disease, and ticks are eradicatable. These are hard species to get rid of. You plug your home, they're back in 6 months. But ultimately, this was going to happen. Fipronil in Mexico is actually dirt cheap, and you can use fipronil as a spot on. It's not cheap here, but it is there. They can spot it onto the back of the dog. It kills ticks. So how to control a large urban outbreak associated with poverty? I don't have an answer to it. Putting this together with our work with Zach, will the tropical tick continue to spread north? Is this a problem we're going to see? We know it's in the United States now. Are we going to see outbreaks like this associated with the tropical tick? If the tropical tick is bringing a more virulent strain of rickettsia, or is a more prolific human biter, that's a problem. How much has hybridization and climate change changed the ecology of the brown dog tick? We don't know. And why so fatal? I mean, you want to say delay in treatment, you know, people delaying because they're poor and they can't afford treatment. Maybe, except I can tell you one thing, that every doctor in Mexicali can recognize Rocky Mountain spotted fever, and most doctors in California could not, right? So it's not that easy. I don't know. And so far, we now have the brown dog tick from Mexicali in culture at CDC, but we don't have the rickettsia from Mexicali in culture yet. We need to, and we will. All right, thank you. We'll get to the last slide, which is a acknowledgement slide. This is our team assembling in the morning, getting ready to go do their thing. I do gracefully, very gratefully acknowledge funding from our CDC border surveillance team, which is called BIDS. UC Nexus funded Zach's work, and then our Center of Excellence, and then the team that was involved in the undergraduate GDB study, and then some of the main co-authors on the Mexico study. And with that, I have my time for a few questions. Thank you very much.